This is the second video for photosynthesis. We're going to look at the game. We've talked about the players in the last video and what they do. Now we're going to see how they all interact. So on this big sheet, big picture is that you have the cell. You have your plant cell. Let me just move one of my little bars up here really quick. All right, and you have the cell membrane up here at the top, and obviously, because this is a plant cell, you can also go ahead and add the cell wall if you want. You don't have to, but you can. And in this cell, I'm just going to show you one giant chloroplast. There's more than just one chloroplast in a plant cell, but we don't need to show more than one for this drawing. And inside the chloroplast, so here's the double membrane of the chloroplast that goes all the way around. Inside that double membrane is called the stroma. All right, so all this is called stroma. We'll return to that later. And then in the stroma, you have these thylakoid membranes. So let me zoom in. All right, so here is your thylakoid membrane. Inside the thylakoid is the thylakoid space. And if you wanted to, if you had a green marker or colored pencil and you wanted to trace this thylakoid membrane in green, remember that's the membrane that has all of the pigments, so that's going to be what looks green to us. That's going to be that membrane. All right, so this video is going to focus on one of the two types of reactions. We're going to look at just the light-dependent reactions today, and we'll do the light-independent or the Calvin cycle reactions in the next video. So remember briefly that light has dual nature, right? It's wave and particle duality for light. So photons are coming in from the sun. Remember, these are not electrons, they're not protons, or they're not neutrons. They are packets of energy. They are discrete packets of energy that are going to come in and hit what's called a photosystem. So I've drawn this photosystem to look like a box. In real life, it doesn't look like a box, but this is just a visual model to kind of understand and simplify what's going on. So these packets of energy are going to come and they're going to hit this photosystem. And they're going to excite electrons in all of these pigment molecules. They're going to excite electrons in all of these pigment molecules. So just as a reminder, electrons exist in orbitals. So here we got orbital levels, and your lower electrons that are in these lower orbitals closer to the nucleus of the atom have lower energy levels, low energy levels. And basically these photons are going to excite these electrons. E with a little negative is a negative charge. That's our symbol for electron. And photons are going to excite these electrons and kick them up into higher orbitals. That's what light is actually doing. So higher energy orbital. And then if nothing happens, if this electron isn't used or even kicked up even higher, it'll go back down to its lower energy level, and you'll see light from that. But that's what the photon actually does. So let's look and see what this photosystem is. So let's turn to the back of your big sheet, and you have a picture of a photosystem here. A photosystem is actually just a protein complex, and in real life it doesn't look like a box. Again, the box is just a model to help understand it better. But it's comprised of all of these different pigment molecules. So all of these circles here are pigments. They can be chlorophyll A, chlorophyll B, carotenes, xanthophylls, anthocyanins, right? All of these different pigment molecules. And remember, all of these molecules are embedded in the membrane of the thylakoid membrane. In the center of this photosystem, of this complex, are two chlorophylls in particular that are kind of special. And they're special because these two chlorophylls will eventually donate electrons that get kicked up into an electronegative reaction center. Sorry, they're in the reaction center. They're going to get kicked up into what's called a primary acceptor. And this primary acceptor isn't anything special. It's just, it's just a protein. It's a very electronegative protein called the primary acceptor protein. By the way, the other thing I want to talk about here, sometimes these pigment molecules as a system, not only are they called a photosystem, sometimes they are also referred to as a light or the light harvesting complex. So you might see that too. All right. So light's going to come in and hit photosystem 2, PS2, and it's called photosystem 2 because it was discovered after photosystem 1. But we're going to talk about it first. And photosystem 2 does a really good job at absorbing wavelength, light wavelength of about 680 nanometers. It has a range to it. It can go down to something like 650 nanometers also, but this is where it absorbs the best wavelength of light. So here come these photons, and they're going to hit, they're physically going to collide with the pigment molecules, and that's going to excite electrons in these pigment molecules. It will kick them up into a higher orbital energy level, and they're going to bounce around kind of like a pinball machine, right? So all these electrons are going to bounce around between all these pigment molecules like a pinball machine, but they're all going to empty, they're going to empty out into the two chlorophyll A pigments in this reaction complex. And it's not just one electron that's doing it. There's hundreds of electrons doing this. So the sum total of all of those electrons empties out into these two chlorophyll A pigments here. And the sum of all of that electron movement is going to kick out one electron. Don't worry about the number of electrons. It's just an electron into the primary acceptor. So really the only purpose of light here is to excite some electrons and kick them into an electronegative protein. That's really the whole goal. And I'm going to go ahead and I'm going to do my best to keep, I was going to say keep electrons negative, but I'm having a hard time without changing everything. So there's our electron. We're just going to have a little blue electron here. Now the electron is going to get passed around like a hot potato, and we've seen this story before, right? While we're at it, let's just go ahead and label this, just so that we don't have to flip the big sheet over again. This was the primary acceptor, and that was a protein, and remember that it is electronegative. And I think we're okay with this over here. All right, so let's see where this hot potato passing takes us. So this electron is going to get passed to the next more electronegative complex. So what you're going to see here, you don't need to know them, but they're included simply because the textbook has them. There's a PQ and a PC. If you're really curious, again, you don't need to know it, but if you're really curious, that's what they stand for. They're there for completion. If you saw this in the text and you were like, well, why are they in the text but not in the big sheet? I didn't want you to be confused, but you really don't have to pay attention to them. The point is the electron gets passed to the nearest electronegative thing. In this case, after PQ, is the cytochrome complex, which is really, really similar to cytochrome in the electron transport chain of cell respiration. And this is a pump. So when this cytochrome complex, this protein complex, has a negative charge pass through it, there's a conformational shape change, and that's going to pump a proton from the stroma into the thylakoid space. into the so only I know I drew two, but you're gonna one electron gets you one proton. So one proton into the thylakoid space. So every time this happens, you get an additional proton. So we're gonna start to pump protons into this thylakoid space. All right, well let's keep going. Next electronegative thing is PC, but we're gonna actually stop here. So let's park our electron at PC. And at this point, what I want you to realize is that all the way back here in photosystem 2, this poor chlorophyll A has a hole. It's missing an electron. The electron has traveled all the way down here towards photosystem 1. It needs that electron back. So we're going to revisit that. So pay attention to that. Look out for that. We'll see where that electron comes from. All right, so we've made our way over to photosystem 1. And what I want you guys to realize is that photosystem 1 is getting bombarded with its own photons. Photons are also hitting photosystem 1. It's not like they're only hitting photosystem 2. So here come the photons. They're hitting photosystem 1. By the way, photosystem 1 was discovered first, so they numbered it first, even though we're talking about it second. 700 is the wavelength of light that it absorbs best. But here come these photons, and the exact same story happens. These photons hit the pigment molecules. That excites the electrons. The electrons bounce around like little pinballs between all of the different pigments that are in this photosystem complex, and they eventually all empty out into the middle where you've got, again, two chlorophyll A's that kick up an electron, right? The sum of all of these electrons bouncing around and accumulating here in the reaction center in the middle result in an electron getting kicked up into this primary acceptor, just like in photosystem 2. So again, if you want to label that, that is a primary acceptor, just like we saw before. And you guessed it, a game of hot potato proceeds. So the electron, again, this is the movement of the electron, so I'll label that. The electron gets passed down to NADP reductase. And I love the name of this enzyme because it tells you exactly what it's going to do. It's going to reduce NADP+. So NADP+ swoops in, says, thank you very much for that electron. I am going to reduce into NADPH, and we'll talk about where NADPH goes in the next video. All right. The hole that this chlorophyll A molecule in the reaction center has, because remember, its electron traveled all the way up to the primary acceptor and then all the way over to NADP reductase, this hole easily gets filled by the electron coming from photosystem 2. So the hole is filled in photosystem 1 using the electron that traveled to it from photosystem 2. But the hole that was in photosystem 2 is still there. It's still there. So where is that new electron going to come from? Where is that new electron going to come from? Because all we've done at this point is pass around electrons to make NADPH. Well, remember that plants are also bringing water up from the roots. And water will diffuse into the thylakoid membranes, or through the thylakoid membranes into the thylakoid space. So let's make a little note of that. Water from roots, and it's entering the space, the thylakoid space, via osmosis. And remember that water is just an oxygen attached to a proton and an electron, and attached to a proton and an electron. So I'm going to redraw the hydrogens as a proton and an electron. There are enzymes in the thylakoid space that cleave water. They break water. They break it into oxygen. I don't know why I keep getting a little box there like that. Yeah, we'll just put it over there. They break it into oxygen, and then they break the hydrogens. They oxidize hydrogen. So they separate the proton from the electron. The proton from the electron. All right, so let's see where all the pieces go. Well, the oxygen diffuses out of the cell. It diffuses out of the thylakoid space, through the thylakoid membrane, out into the stroma, out the inner and outer membranes of the chloroplast, out into the cytoplasm of the plant cell, and then out of the stomata, out into the atmosphere. So this is actually the source of the oxygen given off during photosynthesis. Remember, oxygen is it's an unstable atom, so it tends to combine with itself. So this is going to turn into O2. It'll pair up with another free monatomic oxygen. But this is the source of all of our planet's atmospheric oxygen. All of Earth's oxygen gas comes from this process, whether it's in a plant, a cyanobacteria, or an algae, or protist. All of Earth's oxygen gas comes from breaking water. Every time you breathe and you use that oxygen, in its former life, that oxygen was part of a water molecule, which I think is pretty cool. The electrons, if you didn't figure it out by now, from water are the electrons that replace our missing electron from photosystem 2. So the electrons from water rejuvenate this system. They replenish the source of electrons. The protons stay where they are. The protons chill. They hang out here in the thylakoid space along with the protons that were pumped from the cytochrome complex. So basically what you have here is a high concentration of protons. Some are coming from the water that's getting split, and some are coming from the cytochrome complex, but the pH is actually going to start to decrease because we're pumping in protons here. All right, and the story is actually really similar at this point to what we saw in cell respiration, because here we go. We have ATP synthase. ATP synthase is also in the thylakoid membrane. And the same thing happens in photosynthesis as did in cell respiration. ATP synthase, remember, is a channel protein that allows for facilitated diffusion. I'm just going to write that. This is facilitated diffusion. So these protons are going to diffuse through ATP synthase. That's going to turn a motor, and that's going to provide a mechanical to chemical energy change that attaches a phosphate onto ADP, and there you go. You've made ATP just like you've made ATP in cell respiration. You don't make nearly as much, but you do make some ATP because we will need this ATP for the Calvin cycle, right? So just to apply some of our vocab, remember that the flow of protons through ATP synthase is called chemiosmosis, or chemiosmosis. Chemiosmosis. This combined with oxidative phosphorylation, the pumping in of protons into this thylakoid space, so the combination of those two things is going to get you photophosphorylation. I'm going to type this word out. I know it's a long word. Come on. Photophosphorylation. We're going to phosphorylate. We're going to add a phosphate group to ADP to make ATP, and we're doing that using light energy. So up here at the top is the definition. Making ATP using light energy. you have a spot on the back of your cell respiration big sheet for photophosphorylation. Now would be the perfect time to add this definition to the back of that big sheet. Also, this is completely out of context, but this is a chloroplast. So let's remind ourselves that chloroplasts have their own ribosomes and they have their own circular DNA. Why not? Let's just add that. Why not? They have their own ribosomes and their own DNA. Just to remind ourselves of their evolutionary history. All right, let's talk about what happens to these products, because really all we've made at this point is ATP and some electron carriers, NADPH. Those are the products here. Well, I'm going to go ahead and tell you that the Calvin cycle, there we go. I'm going to write this over here. The Calvin cycle is going to need 12 NADPHs, and it's going to need 18 ATPs. Again, I don't know why the little box is there. For one glucose. So we're going to need 12 NADPHs, and that should make sense because this is C6H12. So, hello, I mean, NADPH, you need 12 of them because there are 12 hydrogens in glucose. 12 NADPHs and 18 ATPs just to produce one glucose. So let's flip over to the back of the big sheet, which for me is just going to be scrolling down to this picture. Because what's going to happen is you're going to need a lot more ATP than NADPH. And if you don't, you're going to have a backlog and the entire system is going to shut down. So let's review really quick, and then we'll talk about how the cell makes that extra ATP so that it can keep making glucose. All right, here's the same thing that's on the other side of the big sheet. Here's photosystems 2, 1. Let's get rid of that. There's NADP reductase. There's the cytochrome complex, or cytochrome. And remember, photons are coming in. Actually, I'm going to go up a little bit and zoom in one more level here. Photons come in. They are going to excite electrons. And remember, that's an electron. The electron is going to get passed to the next nearest electronegative thing, so cytochrome. That's going to cause a conformational shape change in that enzyme, and that's going to pump a proton into the thylakoid space. The electron continues to move into photosystem 1. Right? Now remember, photons are hitting photosystem 1, so electrons move up, just like they did in photosystem 2, into their primary acceptor. The electron moves to NADP reductase, and just like the name implies, NADP gets reduced to NADPH. So that's a cute little overview. And just remember that we also get water. We split water, and the oxygen diffuses out as a waste product, and that's the source of all of Earth's atmospheric oxygen. The hydrogens are oxidized into a proton and an electron. The electron replaces this initial electron lost from photosystem 2, and the protons hang out and accumulate in the thylakoid space. That accumulation of protons diffuses facilitatively through ATP synthase, and we produce ATP, right? This method that we've just gone over is called non-cyclic electron flow. It's also called non-cyclic, oh, it's not going to let me type. Non-cyclic photophosphorylation. I'm just going to abbreviate. Photophosphorylation. Non-cyclic photophosphorylation is another name for it. But what this means is it's linear. It's a linear process. If you follow the electrons, if you follow the electron flow, they're just moving linearly. They're not looping back around and starting again, right? They start as in water, they move up through photosystem 2, through to photosystem 1, and then off into an electron carrier. They don't loop back around and do it over again, right? So this is a non-cyclic linear electron flow. However, that isn't going to produce enough ATP. That's going to produce equal amounts of these, and you need extra ATP. Remember, you need 6 more ATP than NADPH for every glucose. So we need a way to keep an electron in this system without it leaving so that we can use it to make as much ATP as possible. And this is called cyclic electron flow. So if you drop down a little bit, or cyclic photophosphorylation, I'll abbreviate that again here. Photophosphorylation, you can write the whole word yourself. Basically, we're going to recycle an electron. So remember, here, we do have some electrons coming in from water, so I want to include that. Some electrons coming in from water. And again, photons are coming in and they're hitting the pigments, and that's going to excite the electron, and then the electron is going to get passed to photosystem 1. On its way, though, it's going to do a conformational shape change with cytochrome, with the cytochrome protein complex, and that's going to pump protons in. So none of this is any different. And then again, here, photons come in, they're going to excite the electron. And at this point, I'm going to stop, because in order to have this electron cycle back, that means NADP reductase is unavailable to accept the electron. And NADPH inhibits, so you want to draw a line all the way down, NADPH inhibits NADP reductase. So this is a negative feedback. This is the typical negative feedback. As you produce product, as you make NADPH, it inhibits its own production. Because if you make too much NADPH with your electrons, you're going to run out of electrons and creating a proton gradient to make more ATP. You have to prioritize ATP, and that's why NADPH will actually inhibit its own production. So when you've made a lot of that, it turns off this reductase, which means this electron can't get past to reductase. Reductase is out of business, right? So where is the only place this electron can go? It has to go backwards through cytochrome, pump another proton in, and get cycled. Well, then it can just serve as its own replacement, get excited again, go back through cytochrome, pump a proton, replace itself, and do this over and over and over and over again. And this happens repeatedly. This poor electron is stuck in a feedback loop. This happens until NADP reductase gets uninhibited. You have basically enough ATP to join up with NADPH, and then they move on to the Calvin cycle, and that frees up NADP reductase. Right? So the fact that you have this poor little electron getting cycled over and over and over again is why this is called cyclic electron flow. And again, you need cyclic electron flow because pumping those protons into the thylakoid space provides the protons that diffuse through ATP synthase. And that's how you generate more ATP than NADPH. You switch to cyclic electron flow. And cells, plant cells in particular, any photosynthetic cell that has chloroplasts will do this. And if you don't have chloroplasts, you do this through the cell membrane, right? So there's a constant switching between non-cyclic and cyclic flow based on how much NADPH you're producing and how much that's inhibiting NADP reductase and how much ATP you get extra as a result. So that's how the cell kind of guarantees that it has more ATP than it is making NADPH. But again, once you have enough, they kind of all pair up and they all go off into the Calvin cycle. So the Calvin cycle will be the subject of our next video. But before I end this video, let's do a quick summary. Here's the chloroplast. Here's the thylakoid membrane that contains the pigments. You have two clusters of pigments called photosystems, photosystems 2 and photosystems 1. As they get bombarded with photons, they each are going to have electrons that get excited. Those electrons get passed to the nearest electronegative thing. For the electron that started in photosystem 2, it'll get passed to photosystem 1 and have cytochrome complex pump a proton into this thylakoid space on its way. The electron from photosystem 1 goes to NADP reductase and is going to prompt the reduction of NADP into NADPH. All the electrons in this system ultimately come from water and get fed into photosystem 2. Oxygen gets released and the protons hang out in the thylakoid space. Those protons diffuse through ATP synthase to make ATP. So our two products are ATP, well, oxygen is a waste product, but ATP and NADPH. And we'll need 12 NADPHs and 18 ATPs to make one glucose in the Calvin cycle. And to guarantee that we have enough ATPs, sometimes the cells switch from non-cyclic to cyclic electron flow. And we'll talk about the Calvin cycle next time. I hope this helped, and I will see you then.